Welcome to the lecture on demonstrating safety and efficacy for medical devices, an overview of FDA, Health Canada, and EU requirements. Demonstrating safety and efficacy is fundamental to medical device regulatory approval. Regulatory authorities such as the FDA, Health Canada, and EU MDR require a structured and evidence-based approach to ensure that devices are safe and perform as intended. This presentation explores the methodologies, evidence requirements, and strategic considerations for demonstrating device safety and efficacy. Early planning and integration of testing requirements across jurisdictions can streamline approval, minimize delays, and enhance market readiness. We will review key regulatory expectations, best practices in testing, and strategic approaches to align product development with regulatory demands. The objectives of this module are to provide learners with a comprehensive understanding of the key requirements for demonstrating safety and efficacy. Participants will compare evidence requirements across major regulatory authorities, analyze real-world case scenarios, and develop strategic testing and validation approaches. Additionally, learners will gain insight into creating timeline projections for development and regulatory submission, ensuring alignment with regulatory expectations and optimizing market entry. The FDA classifies devices based on risk into Class 1, 2, and 3, determining the level of evidence required. Class 1 devices are low risk and typically exempt from pre-market notification, focusing mainly on general controls such as registration and good manufacturing practices. Class 2 devices require demonstration of substantial equivalence through the 510(k) pathway, involving special controls and additional evidence. Class 3 devices are high risk, requiring pre-market approval, PMA, with comprehensive clinical evidence of safety and effectiveness. Key evidence includes non-clinical laboratory studies, biocompatibility testing, software validation if applicable, clinical investigations, and risk analysis aligned with ISO 14971. Understanding these requirements ensures the testing strategy satisfies FDA expectations while mitigating regulatory delays. Health Canada classifies medical devices into 4 risk classes, with regulatory requirements increasing with risk. Class 1 devices require only a Medical Device Establishment License, MDL. Classes 2 to 4 require a Medical Device License, MDL, with Class 4 requiring extensive clinical evidence. Key evidence requirements include ISO 13485 Quality Management System certification, safety and effectiveness data, risk assessment documentation, and clinical evidence for higher-risk devices. A clear understanding of these requirements allows manufacturers to design studies and compile evidence that satisfies Health Canada while coordinating submissions with other jurisdictions. The EU MDR classifies devices into classes I, IIA, IIB, and 3, with regulatory scrutiny increasing with risk. Class 1 devices are generally self-certified, while classes IIA, IIB, and 3 require notified body assessment. Evidence requirements include comprehensive technical documentation, clinical evaluation reports, post-market clinical follow-up, PMCF, unique device identification, UDI, and robust risk management documentation. Compliance with EU MDR standards ensures the device meets stringent safety and performance expectations across European markets. Biocompatibility testing assesses how device materials interact with the human body. ISO 10993 provides a structured framework covering cytotoxicity, sensitization, irritation, systemic toxicity, genotoxicity, and implantation studies. Material characterization, including chemical analysis, identifies potential risks and ensures compatibility with biological tissues. Proper biocompatibility evaluation is essential for implantable or skin-contacting devices to prevent adverse reactions, satisfy regulatory requirements, and support safety claims in submissions across FDA, Health Canada, and EU MDR frameworks. Mechanical testing verifies that devices can endure the forces encountered during intended use. Strength testing evaluates tensile, compression, and flexural capabilities. Fatigue testing simulates repeated use to identify potential failure points, while durability testing assesses long-term performance and longevity. Comprehensive mechanical testing ensures device reliability, supports safety claims, and satisfies regulatory evidence requirements for higher-risk classifications. For devices marketed as sterile, sterilization validation ensures that the process consistently achieves the required sterility. Methods include ethylene oxide, ETO, radiation, steam, and dry heat sterilization, adhering to ISO 11135, 11137, and 17665 standards. Sterility assurance level, SAL, is typically 10 to the power of -6 for implantable devices, representing a 1 in a million chance of a viable microorganism surviving the sterilization process. Proper validation demonstrates compliance with international standards and regulatory expectations, reducing risk to patients and ensuring market acceptance. Bench testing is conducted in a laboratory to confirm that the device meets design and functional specifications. Functional testing ensures that the device operates according to intended use. Performance testing measures critical performance indicators, and simulated use testing recreates real-world conditions. Bench testing provides essential preclinical evidence of safety and efficacy, supporting regulatory submissions and informing subsequent preclinical and clinical studies. Animal studies are preclinical experiments to assess device safety and performance before human trials. GLP compliance ensures study quality and reproducibility. Model selection must reflect physiological conditions relevant to the device. Endpoints include safety measures, performance outcomes, and pathology assessments. Animal studies generate critical data supporting regulatory submissions, bridging bench testing and clinical investigation. Clinical investigations evaluate device safety and effectiveness in humans. Study designs may include randomized controlled trials, cohort studies, or case control studies. Primary and secondary endpoints capture efficacy and safety outcomes, and statistical planning ensures appropriate sample size, study power, and significance testing. Robust clinical evidence is required for higher-risk devices and is a central component of FDA PMA, Health Canada Class 3/4, and EU Class IIB/3 submissions. A drug-eluting cardiovascular stent is high-risk across all jurisdictions. FDA Class 3 requires PMA with comprehensive clinical trials, Health Canada Class 4 requires extensive clinical evidence, EU Class 3 requires notified body assessment and clinical evaluation. Evidence must include biocompatibility testing, drug release kinetics, mechanical testing, animal studies, randomized clinical trials versus standard of care, and long-term follow-up data. This scenario illustrates the need for integrated preclinical and clinical strategies to meet multi-jurisdictional regulatory requirements efficiently. AI-based medical image analysis software presents unique regulatory challenges. FDA Class 2 requires 510(k) submission with special controls, Health Canada Class 2 requires MDL with supporting evidence, EU Class IIA requires notified body assessment. Evidence includes software validation, algorithm performance testing, usability studies, clinical validation using retrospective datasets, comparison to human reader performance, and risk analysis focusing on false positives/negatives. Software devices require both technical and clinical evidence to demonstrate safety and effectiveness in target populations. Wearable continuous glucose monitors require a comprehensive testing approach. FDA Class II requires 510(k) submission, Health Canada Class III requires MDL with clinical evidence, EU Class IIB requires notified body assessment. Evidence includes biocompatibility of skin contacting components, electrical safety testing, accuracy and precision validation, comparison to reference methods, human factor studies, and clinical validation in the target population. This ensures device performance meets user needs and regulatory expectations across jurisdictions. Development timelines vary by device risk and complexity. Concept development involves market research, needs assessment, and initial design. Design planning establishes inputs, risk analysis, and regulatory strategy. Design development includes prototyping, verification, and design reviews. Verification and validation testing ensures performance and safety compliance. Preclinical animal studies support early safety and efficacy data. Clinical investigations enroll target populations and generate pivotal data. Careful planning of timelines and integration of regulatory requirements ensures efficient device development and market readiness. Preparation for regulatory submission involves compiling a complete technical file or design dossier, clinical evaluation report, and supporting evidence for safety and efficacy. Submissions are made to FDA, 510(k), or PMA, EU notified bodies for MDR assessment, and Health Canada for MDL review. Timely and comprehensive responses to regulatory questions are critical to prevent delays and ensure approval. Coordinating submissions and leveraging common evidence across jurisdictions optimizes efficiency and resource use. Engaging regulatory authorities early in the development process allows clarification of data requirements, risk assessments, and study design expectations. Pre-submission meetings with FDA, Health Canada, and EU notified bodies facilitate alignment on submission strategies, clinical trial design, and regulatory expectations. Early engagement reduces the risk of rework, accelerates approval, and ensures evidence generation meets jurisdiction-specific requirements. A risk-based approach is essential for demonstrating safety and efficacy. Implementing ISO 14971 standards ensures structured identification, assessment, and mitigation of hazards. Risks are addressed through design, testing, and monitoring strategies. Risk assessment informs the scope and intensity of bench, preclinical, and clinical testing, ensuring that regulatory evidence is proportional to device risk. Global harmonization ensures that testing programs satisfy multiple regulatory authorities. Following International Medical Device Regulators Forum (MDRF) guidance and internationally recognized standards like ISO, IEC, and ASTM enables simultaneous evidence generation for FDA, Health Canada, and EU MDR submissions. A comprehensive testing master plan outlines requirements for bench, preclinical, and clinical studies, minimizing redundancy, saving resources, and ensuring compliance across markets. Demonstrating safety and efficacy requires a strategic, structured approach tailored to the device, intended use, and target markets. Understanding jurisdiction-specific requirements, implementing a risk-based testing strategy, and harmonizing evidence generation across FDA, Health Canada, and EU MDR frameworks facilitates efficient regulatory approval. Integrating bench, preclinical, and clinical evidence ensures compliance, reduces development delays, and supports timely market entry of safe and effective medical devices. By planning early and following a comprehensive testing strategy, manufacturers can optimize regulatory outcomes and ensure patient safety.